Assalamu alaikum. My name is Asma Bashar and today I am going to present testing method of cold drinks. Objective: to identify and analyze the major chemical and physical constituents in cold drinks such as soft drinks or carbonated beverages and to determine their safety, quality and composition. Its theory and chemistry. Cold drinks are complex mixtures containing water, sugar like sucrose, glucose, fructose, acids like citric acid, phosphoric acid, and carbonic acid, chlorants, preservatives, caffeine in cola based drinks, and carbon dioxide for fizz. The chemical evaluation of these components helps in determining health impacts, ensuring standardization and quality, checking for harmful additives or adulteration. Parameters to be tested: pH value, carbon dioxide content, which is also known as carbonation test, sugar content like Brix value, caffeine test, phosphoric acid test, Benedict's test for reducing sugars, alcohol content, microbial contamination test, artificial color detection. Key chemical constituents and their chemistry. The component is carbon dioxide. Its chemical nature is carbon dioxide gas dissolved under pressure. Its function is gives effervescence and tangy taste. Next one is sugar, which is like sucrose, glucose, fructose. Its function is sweetness, caloric content. Next one is caffeine, which is alkaloid, and its function is stimulates CNS. Acids like carbonic, citric, and phosphoric. Its function is preservative, tart flavor. Color: tartrazine, sunset yellow, and etc. like synthetic dyes. Its function is aesthetic value. Next one is apparatus. The apparatus that we need to perform this practical is pH meter or pH paper, burette, pipette, conical flask, beakers, test tubes, filter paper, water bath, spirit lamp, refractometer, chromatography paper, distillation setup for alcohol detection, incubator or petri plates for microbial analysis, UV spectrophotometer for advanced caffeine test. Chemicals: distilled water, sodium hydroxide, lime water, phenolphthalein, Benedict's reagent, ferric chloride, iodine solution, silver nitrate, chloroform, sulfuric acid, potassium dichromate, alcohol, microbial growth media. Procedure with scientific logic. First one is pH measurement, which is acidity test. Pour 10 ml of cold drink into a beaker. Insert the pH electrode into the sample. Logic: the pH indicates the hydrogen ion concentration. Soft drinks are acidic due to carbonic acid and added acids like phosphoric or citric acids. Expected result is pH is 2.5 to 4. Next one is carbon dioxide detection, which is carbonation test. Shake the cold drink to release carbon dioxide. Pass the gas through the lime water using a delivery tube. Its logic is the effervescence in drinks is due to carbon dioxide dissolved under pressure. When released and passed through lime water, it reacts to form a white precipitate. Sugar estimation, which is Brix value via refractometer. Clean the refractometer lens, place 2 to 3 drops of cold drinks and close the lid. Read the Brix percent value. Its logic is Brix measures sugar concentration based on the refractive index. Sugar increase the refractive index proportionally. Expected result is 8 to 12% Brix. Reducing sugar test, which is Benedict's test. Take 5 ml of the drink in a test tube, add 2 ml of Benedict's reagent, heat in a water bath for 5 minutes. Its logic is reducing sugars like glucose or fructose reduce blue copper to red copper oxide in alkaline medium. Expected result is green to brick red precipitate, confirms reducing sugars. Phosphoric acid detection. Add 5 ml cold drink in a test tube. Add 2 to 3 drops of AgNO3 solution, which is silver nitrate solution. Its logic is phosphate ions from phosphoric acid from an insoluble salt with silver ions. Expected result is yellow PPT confirms phosphate ions. Caffeine detection, which is solvent extraction method. Mix 25 ml cold drink with 25 ml chloroform in a separating funnel. Shake and let layers separate. Collect chloroform layer, evaporate and observe residue. Logic is caffeine is more stable in organic solvents like chloroform. Evaporation leaves a crystalline residue. Advanced use UV spectrophotometer at 270 nanometer to quantify caffeine. Next one is alcohol detection, which is potassium dichromate test. Distill 50 ml of cold drink, mix distillate with potassium chromite oxide and sulfuric acid. Its logic is alcohol residue orange chromium to green chromium in acidic medium. Expected result is no color change, no alcohol. Artificial colors test. Extract 5 ml drink with alcohol, spot on chromatography paper, use water or alcohol as solvent, run chromatography, observe separation. Logic is dyes travel differently based on polarity and solubility in the solvent, separating them into distinct bands. Next one is microbial contamination test. Pour 1 ml of drink on sterile nutrient agar plates. Incubate at 37 degrees Celsius for 24 to 48 hours. Its logic is if microorganisms are present, they will grow into colonies visible on the agar plate. Expected result is no colonies if properly preserved and sealed. Observation table format. For example, test is pH test, its observation is 3.2, interference acidic. Next one is carbon dioxide test, which turns milky, carbonation present. Brix, 10% Brix acceptable sugar content. Benedict's, red PPT or red precipitate, reducing sugar present. Phosphoric acid, yellow precipitates, confirmed. Caffeine, residue in chloroform, caffeine detected. Alcohol, no green color, no alcohol present. Artificial color, color spots on paper, synthetic dyes present. Microbial test, no growth, microbiologically safe. Applications. Quality control in beverage manufacturing units, food safety for compliance with regulatory bodies like FDA, PSQCA. Research, understanding health implication of drink constituents, education, teaching food chemistry and analytical chemistry, consumer awareness to prevent health risk for adulterated products. Future recommendations. Advanced instrumental methods, HPLC for caffeine sugar profiling, GCMS for trace contaminants, ICP-OES for metal detection. Regulatory policies, strict control on artificial colors and caffeine levels, mandatory display of pH and sugar content on labels, eco-friendly alternatives, encourage use of natural colorants and sweeteners, reduction in phosphoric acid to avoid bone health issues, microbial shelf life studies, conduct time-based microbial studies to set expiry.